Hello, good evening, everyone. So today is apart from the 12 week courses, today is some extra thing which has been arranged for you specifically to address some of the other concepts that is left behind in this course of the omics interactomics course, which we had started with the omics technologies of mass spectrometry, based proteins, and NGS. So what we would do is that today we would be looking at different concepts of the genomics technologies along with the things for the interactomics of which you had gone through for along with that some mass spectrometry based techniques for IP immunoprecipitation and other things. So definitely what we would be doing is today is we'll be looking at the different genomics technologies, some of the repositories and portals, some hands-on and some hands-on on the how to understand and analyze the raw mass spectrometry data if you want to find out and identify the proteins. So what you want to do or what how you would be doing that you can measure or uh, this. So that's why we have started with the omics technologies or the mass spectrometry based proteomics and the NGS. So we will be starting with the genomics. So the mass spectrometry is something. So say if I say so we have proteins. So I am talking about, as you can understand, it's bottom up. As we can understand, bottom up proteomics. So here we do the peptides and try to match with the proteins. So the major difference between the gene level task or the genomics technologies or the proteomics technologies is that the genes or genomes or DNAs can be amplified, right? So it can be amplified. So the technique to understand. So and the major thing is that proteins are expressed or quantified, right? So proteins are expressed or quantified, but genomes is that we have the DNA sequences, gene sequences. So when you want to understand what is a sequence, so in genomics, when we talk, it's about the sequence. When at the transcript level or at the mRNA level, we look at the expression level, and at the protein level, we look at the expression. So this is the major concept or major thing. So here we need to have the sequencing technologies where it is sequenced. So in a way, you can tell that mass spectrometry is also kind of protein sequencing, but it depends on the databases, and it from the theoretical genomics database, it will make the convert translate it into proteins, and from proteins it will convert it to get the theoretical spectra out of it, and from this theoretical spectra, once it you are receiving, you are trying to match it with the experimental spectra that you have observed from the mass spectrometry data with when you go to the finding of the theoretical spectra. So this is a kind of identification and then quantifying of those. But here sequence is that to know the sequences, that what exact is the human genome sequences, and I think you all are aware about the human genome project, which succeeded with sequencing completely human genome, and Till now, certain modifications and all those things are uh, we get it right. So this is what the sequencing technologies then is finally analyzing. So if we talk about the word genomics, so as we have spoken about it in first lecture only, the omics term is something which refers to all, and genomics is something which refers to all the sequences present. So it is like the sequencing technologies where we are having the sequences, and then two. So once we have got the whole sequence done and understand the whole sequence, then we will have to analyze the data and finally interpret the data. So most of the thing is that to know is that what are the possible changes. So what are the possible changes or mutations that is there, or the variations that are present for in a disease condition or a normal condition, and what is receptive. To, yeah, sorry. So finally, you have to interpret the data. So this is what there for the genomics or the genomics based technologies, right? So now we would be. So what is the sequencing technologies, right? So the sequencing methods, of which was one uh, very much used, or the chemical based sequencing methods was, you know, that it was the Sanger sequencing methods, or the uh, for okay. So what did Sanger sequencing methods did? Sorry, as the driver is facing some technical issue. Yes, so we are resuming back. So, genomics, we started with the sequencing method of the Sanger sequencing. So, wow, what is the concept? So, if anyone wishes to say, so you will have the sequence PCR amplified and DNA. So, major thing is that in the proteomics or protein sequences or proteins cannot be amplified as like the DNA sequences or those things, DNA genes can be done. So, what we are doing is that sequencing by, so we are terminating. So this is also known as your, if we talk about, so it is also known as your chain termination method, right? So what it does is that you are mixing DNTPs and fluorescent enable DNTPs. So when you are, so how does the sequence happen? So DNTP is a free uh, hydroxyl group which you can incorporate the next NTP, right? So if you have a DDNTP or dideoxynucleotide triphosphate, DDNTPs or dideoxynucleotide triphosphates, then what it happens is that it had got no free it had got no free hydroxyl group, so then it does not. So what it happens is that it gets terminated at the sequence where that particular thing is there. So now what is happening is that you have the sequence, right? And now if complementary sequence it will be getting added, and if the complementary sequence of the mixture of the DG and TPS come, so then it will have small fragments, and then what you are doing is that you are taking or calculating the sequence. So that was the stronger sequencing with the uh, GD NTPs and with the chain termination method. Now the whole field changed or revolution was revolutionized with the advent of the coming of the next generation sequencing methods, of which Illumina technologies such as MISEC, ISEC 2500, ISEC 3000, ISEC 4000, ISEC X10, ISEC X5, NextSec 550, etc. are some of those methods or some of the instruments that are being used, and it is used is that the sequencing by synthesis approach, right? Is BS or sequencing by sequencing by synthesis, right? So this is what right? So the sequencing by synthesis approach. So what is the workflow? So library preparation. So first you take the DNA fragments and you perform a fragmentation of DNA, which is will have you have 200 to 500 base pair, and then 5 prime and 3 prime adapter are added. So this is known as the process, which is known as your fragmentation. So what this process is here? So that is fragmentation and ligation. So that is your fragmentation. So this first you are sharing of the genomic DNA process. Right? So the fragmentation process, that is the fragmentation of the ligation. So second is the cluster generation or pooling, that is attaching of the random sequence randomly on the layers of the surface closely by matching with the adapters that are amplified. Right? So first is your library preparation, which is fragmenting of your genomic DNA with the 200 to 500 base pair in length, and then 5 prime and the 3 prime adapter are added in a process known as fragmentation, that is the fragmentation and ligation. Then is the cluster generation or pooling, which is attaching your DNA sequence randomly on the length of the surface of the flow cell by matching with the adapters under amplified sequencing DNA polymerase connector primers and fluorescently labeled DNTPs, fluorescently labeled DNTPs, uh, which are added to the reaction. 3 prime protected to ensure one base at a time. It's a signal recorded optically converted to uh, sequencing basis. Demultiplexing, that is the raw sequencing data stored into individual datasets using unique molecular barcode and doing accurate sample identification for the downstream analysis. And then finally, perform the analysis. So So it will be matching interrupters, and then what it will do is that it will amplify it. Then okay, so this is what it is known as your sequencing by Okay, so now is the ion torrent of the thermophysicists scientific. So this is done through the detection of hydrogen ions. So what happens is that DNA polymerase incorporates some nucleotides, which causes pH changes proportional to incorporated bases. So pH changes are some things which are detected as electrical signals, revealing the DNA sequence without the need for fluorescent labels. So it is detecting. So that's one was your SBS approach where fluorescently labeled uh, so laser excited fluorescent signals will be there that which DNT is which nucleotide is getting incorporated and giving the signal. But here what happens is that once it incorporates the nucleotides causes pH changes proportional to incorporated bases, and these pH changes are detected as uh, electrical signals. So the change in as electrical signals and revealing DNA sequence without the need for this fluorescent labels. And then it's a single molecule real-time sequencing, which was sequencing by binding. So it involves observing the incorporation of fluorescent label nucleotides in real time as DNA polymerase synthesis. So DNA strand, as it is also known as your single molecule real-time sequencing, which is that sequencing by binding, where the sequencing involves observing the incorporation of the fluorescently labeled nucleotides, which happens in your real time, and the DNA polymerase synthesis, the DNA strand. This will be incorporated native nucleotides need to cleave fluorescent modifications. So it is just the way fluorescent labeled nucleotides are being uh, incorporated in real time manner as the DNA polymerase synthesizes DNA strand. It incorporates the native nucleotides and eliminates the need to cleave fluorescent modifications. right? So what we have seen that in sequencing technologies like iTorrent, which has been used by the changes of the pH changes, but through the sequencing by synthesis approach, what uses like your Illumina, then you have the specific Byzance smart SMRT system where you are doing the using the real time measurements, which you have the sequencing by the binding and have the fluorescent reliable nucleotides in real time and the DNA polymerase synthesis the DNA strand and it incorporates native uh, nucleotides. Fluorescent modifications. And the next, which is evolving technologies for the Oxford nanopore technologies, which uses nanopore sequencing where single stranded DNA molecules. So the single stranded right? So this single strand DNA or RNA molecules pass through the nanopores, denoting characteristic disruptions in electrical current. So the change in the electrical current used to identify the decipher the basic is real time, like the SMRT or the specific biosensors, which has single molecule real time sequencing, where sequencing observing and in the incorporation of fluorescent level nucleotides in real time as DNA polymerase synthesizes the DNA strand. And this incorporates nucleotides. So similarly in the electrical current. So let us now look at this paper to get a on hand feel about this particular new technology. So this is what it tells about. So this is our nanopore sequencing technology, bioinformatics and applications. So rapid advances in nanopore technologies for sequencing single long DNA and RNA molecules have led to substantial improvements in accuracy, read length, and throughput. These breakthroughs have required extensive development of experimental and bioinformatic methods to fully exploit nanopore long reads for investigations of genomes, transcriptomes, epigenomes, and the epitranscriptomes, and it's being applied in genome assembly, full-length transcript detection, and base modification detection, and in many more specialized areas such as rapid clinical diagnosis and outbreak surveillance. So many opportunities remain for improving data quality and through the base calling methods experimental to particular applications. So nanopore sequencing and its application in basic applied research have undergone substantial growth. First nanopore sequencer, that is Minion on 2014. This technology relies on a nanoscale protein pore or a nanopore that serves as a biosensor and is embedded in an electrically resistant polymer membrane, right? Nanoscale protein pore or nanopore that remains embedded, which remains embedded in an electrically resistant polymer membrane. In an electrolytic solution, a constant voltage is applied to produce an ionic current through the nanopore such that negatively charged single-stranded DNA or RNA molecules are driven to the nanopore from the cis site to the positively charged trans site, controlled by a motor protein that crashes the nucleic acid molecules in the nanopore in a stepwise manner. Changes in the ionic current during translocation corresponds to a nucleotide sequence present in the sensing region. Decoded using real-time sequencing of single molecules, controlling translocation, motor protein as leakage activity, and need to be unbound into a single that passes with the nanopore. A Minion flow cell containing 500 with 4 nanopores in each channel. Nanopores used to sequence DNA or RNA. The wells are inserted into an electrically resistant polymer membrane supported by an array of microscopes connected to a sensor chip. Each channel associates with a separate electrode in the sensor chip and is controlled and measured individually by the application-specific integration circuit. Ionic current passes through the nanopore because a constant voltage is applied across the membrane where the trans side is DNA molecule or an RNA duplicate, then single-stranded DNA with negative charge is ratcheted through the nanopore driven by the voltage. So the nanopore characteristic current change is measured is reflected in the corresponding current at 400 paces per second. Introduction to the technology development of nanopore sequencing and discuss improvements in the accuracy, read length, and throughput. ONT data, we describe the main bioinformatics methods applied to ONT data. We can review the major applications. when it goes to the central research, right? Technology development, nanopore design. The concept of nanopore sequencing emerged in the 1980s and was realized through a series of technical advances in both the nanopore and the associated molecular protein. Yes, with an internal diameter of 1.4 nanometer to 2.4 nanometer lysine alpha hemolysine of staphylococcus aureus. Recognizable ionic current blockades by both a crucial step for single nucleotide resolution and both in the engineering of the wild type alpha hemolysine protein allowed the 4 DNA bases. No complexes were not examined in these reports. Mycobacterium mathis, body name SP, that is similar general diameter of 1.2 nanometer. A key advance in improving the signal to was incorporation of processive enzymes to slow DNA translocation. Superior ratcheting DNA through the nanopore. Indeed, the motor protein provided the last piece of the puzzle in favor to lose demonstrate positive recordings of ionic currents for single stranded that will be resolved into signals and nanopore, alpha hemolysine, and MSPA. In contrast to the previous DNA structure was poorly controlled, the addition of the motor protein reduced the fluctuation in translocation kinetics. Same month, ONT announced the first nanopore device Minion. ONT released the Minion to users in 2015 and commercialized it in 2015. Genier technology nanotag based real-time with sequence and nano is based in novel gen optimal system and one by electronic tunneling technology. So this is going to improvement over your time. Genuinely refined the nanopore and the motor protein using 8 versions of the system to do it, including R6, R7, R9, R9.4, 9.5, 10, 10.3. R6 nanopores have not been disclosed. R9 should notable increase in sequencing yield. Cullinan sigma is dependent growth subunit G from Escherichia coli. Translocation rate of 250 bases per second. 250 bases per second. So, mutant CSGS and new motor enzymes to achieve higher sequencing accuracy and faster sequencing speeds. Single DNA twice. However, the R9.4 have difficulty of sequencing very long overload because of the current signal of zeta across 5 consecutive nucleotides. Nanopore and motor protein, several strategies have been developed to improve accuracy, data, ECTS to multiple times, consensus sequence, similar to the circular consensus sequencing strategy of the single molecule logistical on Pacific biosciences that we have written about SMRT. Early versions of ONT sequencing used a 2D library preparation method to sequence HTS DNA molecule twice and 2 strands of a DNA molecule are ligated together by a hairpin adapter and motor protein guides one strand. Hairpin adapter, second strand, the complement. In May 2017, ONT released the widely method. The hairpin strand is like a separator, special adapter. This special adapter for the high probability that the complement strand will immediately be captured for the same nanopore after the template strand. Similar consensus sequence generated as the 2D library. So one is the one experiment is physically connected by a strand is ligated separately to a special adapter. This special adapter has high probability. The 2D adapter is connected to only one method uses a strand of a DNA ligated and the adapter is sequenced. Accuracy has been improved New base calling algorithm developed for independent research, improved by hidden marker model to 70% and 78% by deep learning, extending the read length. Although the accuracy of N2 sequence is relatively low, the deadline provided by electrical detection has a very high upper bound because of it relies on the physical process of legacy translocation. Reads of up to 2.273 megabits were demonstrated. The 70 read lengths depend crucially on the sizes of molecules. Gravity flow cross magnetic beads, from dialysis and plug extrusion. Each MWDN can also be shared to the desired site based on location, medium distribution, and transposes. Cleavage, however, operate present at small offset the desired site weight decrease in single. Higher efficiencies of both adapter ligation and translocation through nanomoles and long fragments. So that it is an improved sequencing yield. .273 MB, they will yield in few thousand bases. So base primarily improvements in each WDN extraction method size. However, there is a trade-off between length and yield. Sequencing yield of the native RNA molecules. The method requires special library. Primer is ligated. cDNA strand can be synthesized to obtain an RNA cDNA hybrid duplex, followed by ligation of the adapter. Quicker, and this good for on-site applications. Later produces a more stable library for longer sequencing courses and therefore produces higher yields. To the nanopore, therefore, direct sequencing of RNA molecule does not generate a consensus sequence compared to editor and typically of lower average accuracy. Independent research. cDNA sequencing by utilizing existing full-length synthesis method. cDNA without PCR amplification. In addition to sequencing, throughput another important sequencing. Okay, so down the data analysis. So one-fold service continual improvement. In addition to in-house data collections and specific menu into specific analysis, better utilizing the base calling, base modification detection, and post-assembly collection. Other tools is long-range wealth. It's two-layer collection, assembly, and multiple update pass data, but we are also applicable to OMT data. Because don't require high computing for advanced skills. Now is the operating system. Also manages data acquisition and real-time analysis, performs local-based callings, and outputs some binary files in fast-file format to store both metadata and data information. Fact 5 forward distant manner 3 piecewise access versions of Minino output one of the first 5 file and third-party software packages regular quality control format conversion generating fast queue files decodes the current signal of the nucleotide sequence is critical for data accuracy and detection for mismodification. Overall method development for base calling went through 4 stages. Base calling from the segmented current data by HML. Base calling from raw current data using a flip-flop model for native individual nucleotides. Training customized base calling models in 2019. Available software packages and measurements per second, processing the translocation of a DNA on RNA is to a characteristic current shift by multiple consecutive nucleotides marked by the length of the nanopore sensing region. Based on current shift to capture individual signals from each KMR, each current signal contains multiple measurements and the corresponding big variance and duration of the Again, converting raw current into event data would basically inclusive raw current data was just less per square P. DNA data developed, nano raw integrated. The DNA developed was developed as in Polish. Detecting N6 methyl adenosine modifications in iron molecules was demonstrated. It is improving certain sources. Hybrid sequencing de novo. So you can understand that there are many things applications. So you can go through it for understanding it in a much better way. But what the basic is that this is a very new technology which is also undergoing continual improvement of this process. While we need to understand that other technologies which have been improved and which have been in place for quite a long time and quite robust and it's used for the different application methods. But all the analysis methods that we check on are all the sequencing technologies which we do for the genomics, like the sequencing by synthesis or sequencing just like the sequencing synthesis, or if we say that next uh, like the specific that for the single molecule real time. So yeah, by now you are understood that we started with the single sequencing for the chain termination method, where it included the need for getting it into the using different chemicals to convert the fragments, and then finally going for the finding the fragments, which is done by followed by the sequencing by synthesis. And the most popular one which is used is the next generation sequencing methods of the Illumina, which contains like for the library preparation, and then followed by fragmentation by cluster generation or pooling by sequencing, then demultiplexing, and finally alignment in the data analysis. And then you have current torrent, which detects through the changes in the hydrogen ion by which DNA polymerase incorporates the nucleotides, causing the pH changes proportional. Then we have the Pacific Biosciences of BIOS, single molecule real-time sequencing. And then we have the Oxford nanobot technologies or the sequencing methods, and it utilizes nanopore sequencing with single stranded DNA or pass through the nanopores and generates your characteristic disruptions in electric current. It is about the basic business in real time. The major goal and the major things which are there are these technologies. But what and another is 10x genomics technologies, tiny doctors at the single cell level, and the PGI tech solutions. So, but uh, though these are different technologies and are uh, being used at different times, and can be used differently for getting different results. But what is the actual thing that we get by this kind of experiment, where we perform the sequencing and get the results? So single nucleotide polymorphism or SNP are one thing which we look on. So why single nucleotide polymorphism is very important, because you know that the codons are degenerative in nature. So if the certain change in the codons with the mutations over time, if it's accumulated, and if it gets changes at the protein level, it gets translated at the protein level, then what it happens is that its single nucleotide uh, polymorphism might get changed, and that might affect the deleterious effect at the protein level. So if we talk about single nucleotide polymorphisms, so what are different types of SNPs that are available? Like the biallelic SNPs, that two alternative nucleotides present uh, two alternate nucleotides specific to the genome, one individual C T nucleotide in a particular locus. So C is replaced by T, which we have said that biallelic SNP. Triallelic SNP multi three alternative nucleotides often interspecific at a particular locus. One person might have an A, another as G, at another as T. So biallelic or triallelic, and the insertion deletion. So the meaning is that biallelic is that just at a particular position in a locus in a chromosome in the given sequence. If certain individuals have A and certain individuals have T, or C and T, so only two varieties are present. If only two varieties are present across the uh, so two varieties are present. So three alternative nucleotides occurring at a specific genomic position at a particular locus. So one have an E or G and another have an T. So all this will give you the trial equation. So only two varieties or more than two varieties presents. Now the different types of mutations that are there, I think of which you all know, was the missense mutation, single nucleotide changes, another in the protein sequence, alternative proteins. So if there's a change to the substitution of one amino acid, silent mutation, no change due to the degeneracy of the genetic code. Nonsense mutation, that there is a introduces a premature stop codon leading to the truncated protein frame shift, causing a shift in needle and then duplication. So these type of SNPs, after you do the genomics analysis, so you will get the variants by type like this in PSM and PS insertion, deletion, mixed, duplication, and number of effects by impact and the number of effects. First, of which if you do the analysis, you will be getting. So these are some of the various databases like the Cosmic Catalog of Somatic Mutations in Cancer or the and DBSNPs and database of somatic open SNPs and geo exome sequencing project. So this is what I felt of adding for you in the extra. So I'll just give hands on one example of one database that you can try it out. Okay. So this is where you can get about the information for the mutations and different for the pan cancer studies which has been taken initiative of NIH for the TCG or the Cancer Genome Atlas. So explore selected studies. So you can at any cancer you can go. Okay, so so just go. Or what you can do is that just go by, so you go for this and you just query by gene, you can do right? So you can get to know about all the genes and the mutation status. All these things that can be you can get to it. Okay. Okay. So you have any query you can ask, or we can uh, discuss, or then you can end at 8 p.m. Okay. Thank you. I think we can end the meeting and then we will continue with the next week's live session. Yeah. Thank you for joining.